We are recording. This is as usual. If you're trying to get credit for watching the video, every time I write it, I say a screen word. Good grief. Every time I say an attendance word or phrase, write it down, take a screenshot, and then at the end, email that to me, the list of words and a screenshot. And also include one question about the material covered in this video. And it's about 7:50. So if you're watching the video, you can skip ahead about 10 minutes. And that's when the class will start and when you will get your first word for attendance.
Looked like you just woke up.
I wish. It just feels like it. Now I have to wake up at the same time every day to get those kids out the door.
Oh,
And I didn't go to sleep necessarily that late last night. Oh, I'm so tired.
yeah. I'll try to tell you. And I guess probably it happens to tell you what it's triggered, so we should go into the quota fiber turned in age class or got it redone.
Awesome.
Got it microwave on office.
There will be an announcement about abs. See you soon.
I think it was lab 6.
Okay. Photosynthesis.
Yeah.
Good morning.
Yeah.
Good morning. Anybody do anything fun over the weekend? Nope. Neither. Morning.
Morning.
All right, well, we've got a little bit more than 2 minutes. I'll go ahead and start early with the redundant announcements that you guys already get all the time. For example, you can meet me online to get the answers for exam one. No one's taking me up on that. It's a 5% boost to your grade. And you'll need the answers from exam one to study for the final exam. You can also still retake exam two. You can do that until exam three is due, which is on November 9th. The retakes are unlimited, untimed, open book. You can't go wrong because even if you get a lower grade, we'll just keep your original higher grade. So if there's no reason not to retake exam two, just make sure you let me know when you're done so I can adjust your grade. Any questions about exams at all? Even if it's not what I said, just exams in general. All right, everybody's good on exams. Okay. Again, to remind everybody, make sure you check your emails often, your emails and your course announcements. And again, with it being fall and winter's coming, we might have snow days, so make sure you check your email before you come in because I might be stuck. I might not be able to come in which case I'll email you Say, hey, we're meeting online today. Here's the link. So make sure you do that, especially if you're a commuter, right? If you're on campus, it's a little bit inconvenient that you had to walk here and then find out there's no class, but to drive here and realize that would be a real pain. So yeah, make sure you check your announcements and your emails. Any questions about that? All right, office hours today are from 9 to 10, Wednesday from 11 to 12, and Friday from 9 to 12. I'm here to help you, so please take advantage of that. Even if it's something outside of biology, I don't care. Like, you can't be learning biology if you're worried about some math exam that you have. So if you need help with anything, let me know. And if I can't help you, I will. If I can't help you, I'll try to find somebody who can. So yeah, let me help you. Come visit me during office hours. Any questions about office hours? All right, I'm behind on grading. I've got hundreds more students than I've ever had before in one semester. So I'm behind on that, which is not fair to you. So to make things fair, since I'm not grading your stuff on time, I'm not going to penalize you for not turning it in on time. So until I get caught up, so basically, I guess what I'm saying, if you have something that's late, you haven't turned it in yet, go ahead and turn it in because I'm not going to penalize you for it. So until I get caught up with grading, yeah, basically, if you've turned it in before I get around to grading it, then in my book, it's going to be on time. I think that's the only fair thing to do. It's just my other courses are in online instead of in person. So I have to really, really prioritize getting their stuff graded on time. And, because that's the only interaction I have with them is me grading and giving them feedback. Like here, we at least get to see each other every 3 days. So they are my priority as far as grading is concerned. So you're like my little stepchildren. So I apologize for that. Any questions about that? All right. Attendance, as always, is mandatory. Act turned in. Participation is graded. So, if you miss a day, even if it's an excused absence, you need to go back and watch the video. And as usual, like we've been doing recently, even though you're in person, you need to write down the attendance words and email those to me. And of course, the last word for attendance is going to be asking me a question about the material covered in this lecture. So keep that in the back of your mind. All right. Let's get started. Officially, you get started. So speaking of attendance, first word for attendance today is Scott Weiland, or Scott, let's just do Scott. Scott Weiland's birthday, the lead singer of STP. And speaking of attendance, let's take it. Brumfeld is not here. Champion is here. Gross, probably online. Gunther is here. Randall Hill is here. The other Hill is not. Jackson, not here. Keys is here. Perez is not here. Sigmund is here. Slaughter is here. Smith is here. Stover is here. Tompkins is here. Vanessa is not here. Watson is not here. Young is here. Zweidak is here. Lawrence is not here. Wilson, not here. That's it. Did I miss anybody? Okay, nice and easy. Let's get to it. Does anybody have any questions or your own announcements? Any fundraisers, anything like that the rest of us need to know about? All right, let's move forward then. So to remind you, big picture here, this chapter, well, this portion of the chapter is all about this idea of going from DNA to RNA to proteins. And like I said, I suggest memorizing the big picture before we start adding the details, right? So DNA to RNA to protein. Then we talked about how going from DNA to RNA is called transcription, and going from RNA to protein is called translation. Then we started adding the details about transcription, right? And that's where we're at right now. We're looking at the details of transcription. So now I think we left off on Friday saying transcription is broken out into 3 stages. There's initiation, elongation, and termination. And you need to know them. And I'm hoping this is easy because it's in the name, right? So you need to know what order those are in. Obviously, the initiation is going to be first because that's what that initiates. Obviously, termination is going to be last because that's what it means to terminate, right? You end something. So therefore, by process of elimination, RNA elongation must be in the middle. So any questions so far before we go in and look at these details? All right. So it does get a little bit trickier. For example, you do need to know this term, promoter. This is the DNA sequence where the start transcribing signal is, basically the code that says, hey, this is where it starts. So to remind you what we talked about last week, and hopefully you didn't forget it over the weekend, I wouldn't blame you if you did though. We said that the enzyme that does this, that reads the DNA and builds the RNA, is called RNA polymerase, right? So that's a reminder, a refresher. So you already knew that, but now I'm telling you where it attaches to the DNA, and you need to know that for the exam. It starts on a place called the promoter. And if you've done those videos in the for the lab, I forgot which lab, but one of the videos talks about that. And it actually gives you the code. Not that you need to know it, but the video gets pretty specific and tells you specifically which letters there are for the promoter. But yeah, you just need to know that's where it starts, the promoter. Obviously, it's going to be located at the beginning of the gene. It wouldn't help if you had the promoter in the middle of the gene, because then you wouldn't, the RNA polymerase wouldn't read anything behind it. So it needs to be at the beginning of the gene. So in short, transcription is simply when the RNA polymerase attaches to the promoter. That is initiation of transcription. Initiation, that's what happens during initiation, right? The RNA polymerase attaches to the promoter. That's it. Nice and simple. Here's a picture. It's not from your textbook. It's from a different textbook. So you don't need to worry about this 5' and 3' and upstream and downstream. All that would be important if this was a more advanced course, but it's not. So there we go. Any questions about initiation of transcription? Okay, so I'll go ahead and tell you now. It will get a little bit more difficult, because when we talk about translation, the next step, right? When we start talking about translation instead of transcription, there's also 3 steps. And it's also initiation, elongation, and termination. However, the terminology used when describing how that starts is a little bit different. So honestly, that's probably going to be the hardest part. Well, not the hardest part, but one of the hard parts of the exam is keeping those separate, right? We're trying to remember which one was initiation and which one was, which one was transcription and which one was translation. So yeah, keep that in the back of your mind. But yes, in transcription, it starts in the promoter. So any questions? All right, second phase. This one's pretty simple too. It's in the name. Elongation, right? It's the elongation of RNA. That's just when the RNA is getting longer. So in initiation, RNA polymerase attached to the DNA, so it can open it up and read the code. And then elongation is simply as it moves down the DNA molecule and opens it up and reads the code, like we talked about last week. It's bringing in these nucleotides, right? It'll hydrogen bond those nucleotides there, and then covalently bond the nucleotides to each other. It's pretty simple. Well, it's pretty complicated, but this description is pretty simple. What you need to memorize is pretty simple. So anyway, yeah, as this RNA polymerase is moving down the DNA, obviously the RNA itself is getting longer and longer until it's finally done, and that'll be the next step. So we'll talk about that. I guess the only other thing to point out is, again, like I said last week, this RNA polymerase is opening up the DNA so it can read the code. And then when it's done reading the code, it closes back. And again, the videos that you've watched describe this and actually describe it in more detail. So when you're studying, keep in mind, like, those videos added more detail than you need to know. Like, for example, like we said last week, RNA polymerase, this is an oversimplified explanation, because RNA polymerase itself is a team of molecules. There's a team of enzymes that have different jobs to do. Like, opening up the DNA is one job. Closing up the DNA is one job. Those are all kinds of different stuff. Primates, right? So there's things that have to be, these bases have to be primed to do these things. But again, you don't need to know those details. You just need to know these details. So any questions about elongation? All right, nice and simple. The last step is termination. Again, it's in the name. Hopefully that's easy to remember. And where does it stop? So we know it starts at the promoter. Where does it stop? It stops at the terminator. That's nice and easy, right? Starts at the promoter, ends at the terminator. And just like the promoter is a certain code in the DNA that says, hey, RNA polymerase, this is where you attach. Same thing here, right? It's a code in the DNA that says, all right, RNA polymerase, you're done. Your job here is done. You can release and go do something else. So any questions about that? It's really simple, not much else to say. All right. Good timing. The next word for attendance. So since we have somebody here to remind you, since you just came in, you're gonna have to send in the attendance word at the end of the day to get participation points. And the next word for attendance is beer. Today, for some reason, is National American Beer Day. Beer is the next word for attendance. Anyway, any questions about transcription? Excuse me, about termination, or for that matter, now that we're done talking about transcription, are there any questions about transcription at all? I'll show you a picture of the whole thing. I think this is from the old textbook and not yours. I don't know. Anyway, it's a really does a great job of visualizing what we're talking about. Here's the promoter, right? That's where the RNA polymerase knows to attach. That's what initiation is, when the RNA polymerase attaches to the gene, to the DNA. Elongation is it's simply moving along, right? It's moving along. It's reading the code. It's making new RNA based on the code that's written in the DNA. And then eventually it hits the terminator. All the parts go separate. And that is the end of transcription. Again, this is a very oversimplified version. If you were to look into it, it's much more complicated than this, but these are the basics. And again, like I said at the beginning of the chapter, you can't really understand how viruses work or how mRNA vaccines work or true gene therapy, how that works without this. So if you ever have to do some research, or more like looking stuff up, you don't have to start from scratch, right? You'll know the basics. Anyway, if there's no questions about that, we'll move forward. If you download the PowerPoint, you can watch this little animation that really shows what we're talking about. And here we go. There's one little step though. Like I said, the big picture here, this second main bullet point of the chapter is all about going from DNA to RNA to protein, right? And we're done talking about going from DNA to RNA. But before we can talk about going from RNA to protein, there's this little middle spot here. It's called the processing of eukaryotic RNA. So in other words, we're done talking about transcription. So we made the RNA, but the RNA is not quite ready to be translated and translated into protein yet, right? We have to process it. So that's what this part is. So to remind you, again, this is the processing of eukaryotic RNA. So eukaryotes are things like us, right? Animals, plants, protists, things like that. We're not talking about bacteria. We're talking about more complicated life forms. That's what we're going to talk about. We're not talking about prokaryotes. I think your textbook might even talk about it. I don't remember. But if it does, you don't need to know about it. I'm just pointing it out here that it's different from them. It's different for prokaryotes than it is for us because we're just more complicated, right? So for them, as that DNA is transcribed, and you have that RNA elongation, immediately it's ready to go, right? Immediately, that RNA that it made is ready to be translated. There's no nucleus, right? So it can just happen right away. But now here's the new term that you need to know. First time I mentioned this, so I've said RNA a lot, right? This chapter I've mentioned RNA many, many times. But now I'm going to get specific because there's different types of RNA, and you're going to need to know the different types. There's a lot of different types of RNA, but you're going to need to know 3 of them. This is the first one, mRNA. This is the messenger RNA. This is the molecule that will eventually, when we talk about translation, will be translated into protein. That's the stuff in an mRNA vaccine. So it's RNA that's going to be translated into a protein. That's basically how mRNA vaccines work. Specifically for the COVID-19 one, to keep it simple, simplify it, there's a code that says build this protein. And it's a harmless protein, but it's the protein found on the outside of the COVID-19 virus. So the virus itself is dangerous, but that little protein is not. So that's why it was such a breakthrough of a vaccine. That way we can just manufacture that protein, and then your body recognizes it and says, oh, that's not good. That way when COVID does enter your body, your immune system has been trained to fight it. Anyway, that was a tangent. I'm not going to ask you any questions about that on the exam, but you definitely need to know what mRNA is. You're going to need to know the difference between mRNA and the ones I'm going to teach you later. So again, we are eukaryotes, right? We have nucleus, right? And that is where transcription happens, but not translation, right? So before this RNA can leave the nucleus, it has to be modified. And another thing, speaking of viruses, and we'll talk a little bit more about this later in the chapter officially, your body, your cells have a way of defending themselves. So viruses, basically, as you're going to learn later, they're either RNA or they're DNA. But that's mostly it. That's how they work. They insert their genetic information into your cells so your cells can follow the code. So what happens is your cells are on the lookout for foreign DNA and foreign RNA, and your cells should generally destroy foreign DNA or foreign RNA. And this is one of the reasons that this has to be modified before it can leave the nucleus, because outside of the nucleus is a very dangerous place for nucleic acids. So that's why we have to do this. And here we go. One of the modifications is something called cap and tail. You need to know this, cap and tail. It's where there's an addition of extra nucleotides at the ends of the RNA. Right? So we have this one long strand of RNA, mRNA. So we add a cap to it, we add a tail to it. This helps protect it from getting attacked. Like I was saying earlier, it's a dangerous place outside of the nucleus. But not only does it do that, but it also helps something you're going to learn about this later. There's something called a ribosome that does the translation, where it reads the code and makes the protein. So that ribosome needs to be able to recognize it and say, oh yeah, that's my thing right there. So that's what cap and tail does. Now you see here, this picture is not from your textbook. I don't think your textbook mentioned the fact that the tail is a poly A, meaning it's just a bunch of A's repeating. And that's on the 3' end. The cap is on the 5' end. You don't need to worry about any of that. You just need to know that cap and tail is one of the things that happens before the mRNA can leave the nucleus. So any questions about that? All right, for extra credit, you can look up some details about this. Like I was just said that we added nucleotides to the ends of both of the strands of RNA. But what did that? Like obviously some enzyme, some cellular machinery must have been responsible for that. So you could look that up if you want. You can either just send it to me in an email. It's like, oh, here's the answer. Don't forget your sources. And that'd be 1 point. Or you can write a paper about it, in which case the point value would be based on how long your paper is. And of course, how well written it is. Anyway, any questions about cap and tail? All right, this one's a little bit more complicated. Splicing. Splicing is another type of modification. This is where we basically remove, I hate to use this word because now we realize it's not what I'm about to say is not exactly correct, but it's a great way to think of it when you're studying for an exam, right? So what this is doing is removing the junk DNA. And again, I don't like to call it junk because it does serve a purpose. You can look it up if you want for to write a paper, for extra credit, look that up if you want. But yeah, we're taking out the junk RNA. I said DNA at first, that was an accident, I'm sorry. We're taking out the junk RNA, splicing it right out. In this picture, I don't remember if this is from your textbook or not, but it's a really good, simple explanation, right? So you can see these parts, these purple or these red parts. It's actually a really bad picture now I'm looking at it. But anyway, here's where it gets a little bit complicated. The part that's taken out, the junk, if we want to call it that, that's called the introns. And the parts that's being kept are called the exons. That always confused me as a student because to me, intron sounds like it's in, so we're keeping it, and exon sounds like we're getting rid of it, it's exiting, but it's the opposite of that. So the way you need to memorize it is the intron was, let me back it up, I should flip those, that might help. The part that we keep is the part that we're encoding. That's the part that's being expressed, hence the word exon. I wish somebody would have told me that when I was a freshman. But yeah, that's what exon means, right? That's the part that's expressed. So hopefully that'll help you remember. The part that's expressed, obviously, is the part that you're going to keep, and that's the exons. The other stuff that we get rid of, it's the in-between stuff. So hopefully that'll help you memorize the difference between the two. There will be test questions on that. So again, exons are the parts that are expressed. That's the stuff that's kept. Introns is the junk in between that we get rid of. That part's pretty simple, right? Again, 5' over here, 3' over there. You don't need to worry about that. You just need to know which parts stay, which parts go. Any questions? Okay. Well, yeah. So that's basically everything I just said, but now it's in writing, right? The introns are removed, the exons stay, they're put back together. But just like I said with cap and tail, notice I didn't give you any details. Like what enzyme did all this? But, like what's the cellular machinery that's doing this? You don't need to know that, but you can look it up for extra credit. Any questions so far? All right. Now, this is actually pretty significant, biologically speaking. Like, because you might just think, okay, well, big deal. We just got rid of some in-between stuff, and now you're just left with a short mRNA molecule. What's that matter? But it serves a purpose. And this is the purpose. Humans have about 21,000 genes, right? But we produce many more than 21,000 types of proteins. So that's got to make you wonder, right? If genes are instructions on how to build a protein, and we only have 21,000 sets of instructions, then how do we have more than 21,000 proteins? This is the answer to that. This is what RNA splicing is for. We can take one gene, and depending on how we splice it, we can come up with different proteins. And we'll talk about that in a second. This slide is out of order. I should have showed you this slide first, and then talked about that. But anyway, sorry about that. So before we talk about what I was talking about with those 21,000 genes, let's back up. This is the basics of what I was showing you earlier, right? We've got exons and introns written into the DNA. It's transcribed. So now we have the exons and the introns written into the mRNA. We remove those introns and splice those exons back together. After, of course, we've added the cap and tail. Now this thing is ready to leave the nucleus. But anyway, again, so that's just a visualization of what we were talking about earlier. Now, going back to this discussion, where we're talking about this RNA splicing and how it allows us to have more than 21,000 proteins, it's something called alternative splicing. This picture is definitely not from your textbook, but it's a great visualization, right? So here we have this strand of DNA. We have the exons labeled, right? There's 4 exons. Notice that some of them are closer together, some of them are farther apart, some of them are larger, some of them are smaller. That's the way it works. So anyway, we have those exons in the DNA. That's been transcribed. So now we have this RNA that also has those exons and the introns that aren't even labeled, right? And then we have different ways of doing it. So you can see this is the typical way that I showed you, right? We got rid of all the introns and brought it all together, right? So now we have exon 1, 2, 3, 4, and 5 all together. And this is what the protein looks like. Now, granted, you guys are 100 level non-biology majors, so that picture probably doesn't make sense to you, but that's okay. It doesn't have to. But the point is, look at protein A versus protein B versus protein C. Even though those probably don't make sense to you, you can still look at them and be like, oh yeah, those are different, right? They're different from each other. They have a different shape. And if you remember way back when I first started telling you about proteins, when it comes to proteins, it's all about the shape, right? So obviously these things are going to have different functions because they have different shapes. But then again, they also have some similar shapes too. So you can probably deduce that the functions are probably similar but different. Even without knowing what protein A and B and C are, you can probably guess that their functions are similar but different. But anyway, that's this translation, right? That's that version of splicing where all those introns are put together. In this version, we have exons 1, 2, 4, and 5, right? So in this case, we left out exon 3. Again, they got a completely different protein. In this case, we have exons 1, 2, 3, and 5. So in that one, we left out exon number 4. So you can imagine, too, this is a very simplified version, right? There's usually probably more than 5 exons, as a matter of fact. That's something else you can look up for extra credit. Like, how many exons does a typical gene have? And I bet if you look that up, the same website will maybe also answer what's the longest, or which gene has the most exons, which one has the fewest. I might tell you that. But anyway, yeah, the point here is, again, with this alternative splicing, there's different ways of putting things together. Again, we have 21,000 genes, but we have many more proteins because we put them together differently. So any questions about that? All right. Obviously, if you're studying the PowerPoints and you're looking at this picture, there's nothing to memorize here. Don't think I'm going to give you a question like, hey, remember that one slide where I showed you alternative splicing? Which exon was left out in the last one, right? They're not going to do anything like that. The closest thing, I don't even think I would do this. I might give you a question where I show you this picture and say, in protein C, which exon was left out? And just look at the picture and be like, okay, 1, 2, 3, 5. Oh yeah, exon 4 was left out. But I don't think I'm going to do that either. This is not middle school. I think that question would be too easy. Anyway, any questions about the processing? Because we're done talking about processing. We're done talking about this in-between part of transcription and translation. At this point, the mRNA that we're talking about, it's ready to go. It's ready to leave the nucleus and be translated. So any questions? All right. Here we go. Let's talk about it. And again, this is from the other textbook, the old textbook. So if you're following along in your textbook, as you should be, right? You should be reading your textbook. It's going to look a little bit different. And I love the old textbook, what they do here, because before we even talk about the process of translation, we're going to explain the parts. because translation is a little bit more complicated than transcription. So we're going to explain the parts, the cellular machinery. Then once you're familiar with all the parts, then we'll talk about how they're all coming together. So the first very obvious important ingredient is the mRNA, right? That's what we just got finished talking about. Talking about transcription, where the RNA polymerase read the code on the DNA and made an mRNA molecule, which was then, modified so it could leave the nucleus. So that's obviously the very first ingredient. Then, this is out of order too. It should be done here. The machine that's actually going to do the translation, that's called a ribosome. So the ribosome, you need to know that for the exam. Ribosomes are the thing that's actually doing the translation. In a sense, you could almost think of it like the RNA polymerase of transcription, right? RNA polymerase is the thing that in transcription that's reading the DNA and making that mRNA. Well, this is the thing that's reading the mRNA and making the protein, the ribosome. You need to know that. Of course, it's going to require a little bit of energy. It's usually ATP. Sometimes there's others. You can look that up for extra credit if you want. What's the other source of energy? Or specifically, how much energy does it take to do this? All these things you can look up for extra credit. But here's where it gets a little trickier. Like I told you earlier, there's more than one type of RNA. You're going to need to know the difference between them. So I'm introducing you to the second one right now. tRNA, transfer RNA. Hopefully you can remember it's called transfer. And once I talk about how it works, hopefully you can remember the difference between the two. I think messenger RNA is easy to remember because it's the messenger. It's like, hey, here's what the DNA code is, right? It's the bringing you the message. So, any questions so far before we talk a little bit more about this transfer RNA? Okay. So, obviously, this whole process requires an interpreter, so to speak, right? Because we're talking about translation. So, we're going from one language to another. We're going from an amino acid language, the nucleic acid language, to an amino acid language, right? We're going from nucleotides to amino acids that are put together to build a protein. Right. So as you already know from last week, this interpreter will convert those 3-letter codons into amino acids, like we already talked about, right? You saw that big wheel, and you can say, all right, C, G, A, and then it will tell you what which amino acid, right? So this is what's doing it. So it's the transfer RNA, in a sense, is kind of the interpreter, because the transfer RNA has a specific code on it for a specific amino acid. So as you already know, there's 20 amino acids. So there's a specific transfer RNA for each one of those codes. So it's the thing that reads the code on the mRNA and says, oh yeah, that's me. And it's the thing that reads the code on the amino acid. Not the code, but it has its own specific amino acid. It would almost be as if each one of you, like if we were to do a demonstration, it would give each one of you a specific amino acid and a specific name. And we would read a code. I'm like, all right, someone please bring me the leucine. And you're like, oh yeah, that's me. I'm that amino acid. And you'd be the transfer RNA and I'd be the ribosome. And you would come over and you would bring me the leucine and I would hold it. And then I'd read the next code. I'm like, all right, oh, I need another leucine. And then the same person would come and give me their amino acids. I put them together. And then I'd read the next code. I'm like, all right, now I need, an arginine. And then one of you who carries the arginine, that's your job, right? So you would come and you would bring it. I would take it and attach it to my longer growing strand of amino acids. That's how it works. It's the tRNA that's doing the interpretation because, again, it's the tRNA that has its own specific amino acid and it reads a very specific code that says, hey, that's me. That's my code. I need to bring in my amino acid. So, any questions about that? All right, so to help you remember that for the exam, again, this is the thing that's transferring the amino acids to the ribosome, right? This is the thing that's transferring those parts. Unlike the messenger RNA, which is just bringing the message, right? This is the thing that's transferring the amino acids. It's not that complicated. It's just that later, when you're studying and trying to memorize the difference between the different types of RNA, that's where it gets a little bit complicated. It's not that I'm sure you probably have no problems understanding how it works. It's just memorizing those different names. So anyway, any questions about that? All right, let me give you a little bit more information than you need to know here. But it's made up of a single RNA strand. This is what it looks like, which is odd, right? Because if you look at it, it looks double-stranded. It's not that it's double stranded, it is single stranded, it's just kind of folded up on itself and made that shape. That is very important for people studying this at a higher level, but for us, it's just trivial. So yes, it does twist and fold up on itself. It does have a very specific shape. The important part to us, like we said, is it has a code. It has its own specific code that it recognizes on the mRNA to say, oh yeah, that's me. I need to bring in my amino acid. But yes, again, for the exam, I would say this slide is completely unimportant. But I wanted to bring it up anyway. Your textbook did, so I wanted to as well. Matter of fact, one of your classmates asked me a question. One of the questions was, you know, at the when they sent in their attendance boards, one of the questions was, what stuff that you mentioned today will be on the exam? I said, well, maybe you would agree, right? I usually tell you, so it's a good time to remind you. If something's going to be on the exam, I usually tell you, and something's not going to be on the exam, I usually tell you. So yes, this stuff not going to be on the exam. Any questions? All right, this probably won't be on the exam either, but I'm also going to get very specific here. I showed you that picture, that last picture at the bottom was that code, right? That 3 letters. What we call that is an anti-codon, which kind of makes sense, right? You already know about codons. Codons are the code written in the RNA. It's just 3-letter words, right? So the anti-codon is just what reads it. And it also, again, I'm not going to go into this much detail on the exam, but if the DNA code, for example, is, I don't know, CCG, right? That would be the DNA code. The RNA code then would be the opposite of that, right? It would be what goes with it. So that would be GGC. So then the anti-codon, I'm just going to abbreviate it, AC. The anti-codon is just the opposite of that. So it's basically the same as the DNA, CCG. They're all complementary to each other, is basically what I'm saying. And again, for the exam, I'm probably not going to mention anything about anti-codons. But when you're reading the textbook, as I hope you are, I want you to be able to understand what you're reading. So I wanted to explain it. Anyway, any questions about the anti-codon? All right, here's a better picture for a 100 level course. This is from the old textbook, I think. But you can see here, this is a very simplified version on the right. And that's the way I think it should be. Like, yes, all these things are folded up on each other with hydrogen bonds, and it has a very specific shape, but who cares? For us, this is just a great visualization. It's kind of got a long shape. You got an anti-codon on one end, and on the other end, you have the spot. Excuse me, good. Where the amino acid attaches. So, any questions about that? So again, to remind you, there's 20 different amino acids. So each one of these tRNAs is very specific, right? It has its very specific anti-codon with a very specific amino acid. One of the 20. So any questions about tRNA? That's it for the discussion on tRNA. All right, so I've already introduced you to this other one, but let's talk about it in more detail. Ribosomes, right? This is the thing that's bringing all the stuff together. So the mRNA that I talked about, the tRNA that I talked about, right? It all comes together with the ribosome. This is the thing that's actually making the polypeptide, right? So remember, a polypeptide is just a bunch of this long string of amino acids. So like I was saying in that demonstration, if I was saying that each one of you was a tRNA and I said, hey, bring me a leucine, and someone brings me a leucine, that's the ribosome, right? The ribosome sitting there and it's saying, all right, well, bring me tRNA, bring me this one, bring me this one. And it gets it and it puts them together and just makes this long strand of amino acids. Now, I'm not sure if I'm going to ask you questions on this, so might as well talk about it just in case. But technically, a ribosome is made up of 2 parts, 2 subunits. There's a small subunit and a large subunit. The small subunit is where the mRNA attaches, and the large subunit is where the tRNA attaches. I'm almost definitely not, again, I wrote a little star next to it to remind myself of this. If I ask any questions about it having 2 parts, that'll be it. I'm not going to ask you to memorize the fact that the mRNA attaches to the small part and the tRNA attaches to the large part. But you do need to know there are 2 parts, so you might need to know there's 2 parts. But when I show you this next picture, it should be easy for you to memorize which part goes on which. Because when you look at it, it's like, oh, that's just kind of the way it has to be. So let's take a look at this next picture of what we're talking about. Here's a ribosome with its 2 parts, right? You've got a small subunit and a large subunit. You can see here, obviously, it would make sense that small part is where the mRNA kind of attaches. In the large part, because remember, we've got those tRNA that are long, right? So it would make sense that you would need the larger part to accommodate for those tRNA. Now I will point this out too. This version, and I think your textbook too, only shows a P site and an A site, which you're going to learn a little bit more about later. But in all actuality, there's a third site that these textbooks don't even talk about. So you don't even necessarily need to know about the P site or the A site. I'm not going to ask you any questions. I'm going to teach you what happens at these different sites. But I'm not going to have any questions. I say, which one of these things happens at the A site, or which one of these happens at the P site, or this thing, does it happen at the A site or the P site? Right? I'm not going to do that. So once we talk about translation, right now, again, we're still just talking about the parts. Once we talk about how it works later, again, you're going to see what happens in these different sites. But I'm not even going to mention it. I'm not going to say the P site or the A site again. I'll just say, right here is where this happens, and then it moves over here, and this thing happens, right? So for this exam, you do not need to know the difference between a P site or an A site. For extra credit, you can look up the other site. What is the other site? What does it do? Anyway, any questions about the ribosomes? Or for that matter, any questions about any of the parts? Because now we're done talking about the parts. We're done talking about the mRNA and the ribosomes and the tRNA, and the amino acids, right? You know all the parts. Now we're going to talk about how they all come together. We're going to talk about the process, if there are other questions about the parts. Okay, let's talk about the process then. This is nice and easy in a sense, because like I said, you need to know for transcription, what's the last one we talked about, it goes initiation, elongation, termination. So the good news is it's the same thing here. So if you memorize the one for transcription, at least as far as nomenclature is concerned, you're good for translation too. Same 3 words, right? It happens a little bit different, obviously. And we're going to talk about that, but yeah, same 3 words. Any questions so far? All right. So let's talk about the different parts, just like the different initiation, elongation, termination. So here we go. Initiation brings all the parts together, just like initiation of transcription. Of course, in that sense, it was a lot easier. The parts were the DNA and the RNA polymerase that attached to the DNA. That's nice and easy for transcription, right? Here, we've got the mRNA that's going to be attached to the ribosome, and that tRNA comes in with the first amino acid. And I'm going to show you a picture of it. Before I show you a picture, are there any questions with what's right on the screen? All right, this is the wrong picture. I guess it's just a good reminder of what mRNA looks like. So again, you've got the coded stuff there, and you've got your cap and tail, whatever. But here we go. And this picture is not from your textbook because there's the other site that I was talking about. But anyway, you need to know for initiation, in short, again, all the parts are coming together is what's in short what happens. Now, where does that first tRNA come? Where does it attach? And that is called the start codon. So in a sense, the start codon is the promoter of translation. Remember, because that's going to be a test question. For transcription, where does it all start? Where does initiation start? For transcription, it starts on the promoter. For translation, where does it start? It starts at the start codon. So that tRNA comes in, that first tRNA comes in, and technically, you know what, I'm not even going to get this detailed either. This picture is very detailed. But technically what happens is you've got this small subunit of the RNA, of the ribosome, small subunit, mRNA attaches to the small subunit, that first tRNA comes in, like, oh, that's me, I'm the first one. It's always that one, by the way. It's always AUG, it's always MET. But anyway, you don't need to know that. That's technically how it happens, right? mRNA first, then the tRNA, then the large subunit. But for the exam, you don't need to know what order it happens. You just need to know that all the parts that we talked about come together, and that's what initiation is. And that very first starting spot on the mRNA is called the start codon. Pretty simple, at least the way we're doing it here, describing it here. So any questions about this before I show you a different picture of the same thing? All right, this is a more simplified version, but yeah, same kind of stuff, right? You got the small subunit, the mRNA attaches to it. That first tRNA comes in, again, always with the MET, right? Always the UAG codon, right? Then finally, the large subunit comes in, and there we go. Now we're ready. Now we're done with initiation. All the parts are together. Now notice that this tRNA is in the P site, not the A site. Now, again, I'm not going to ask you about the names of them. I won't even ask you like, hey, does it come in on the left side or the right side? Doesn't matter in this course. But later, when we start talking about the next step, it will be important, not for the exam, but to understand how things work. It will be important to know that this first one's here, and we have this open one, right? And as you're going to learn later, something's going to come in, and things are going to rotate. So yes, for the process, it's important that that thing is in the P site. For the exam, it's not. I'm not going to ask you that. But for you to understand how it works, that will be important later. So any questions about this picture? Download the PowerPoint. You can watch this animation that shows what I'm talking about. So that's it for initiation. Next on to elongation. Again, the name describes what happens. It's pretty simple. You're elongating this chain of amino acids, right? You're elongating the polypeptide. You're building the polypeptide, building the protein, all these different ways of saying the same thing. We are adding amino acids one at a time, right? It's just a strand of amino acids, one linear string of amino acids, and that's all we're doing. For elongation, we're bringing in different amino acids. What makes it a little bit more complicated is that this is broken down into 3 steps. So all the translation is broken down into 3 parts, right? Initiation, elongation, termination. And now I'm telling you, elongation itself is also broken down into 3 steps. So I'm going to talk about this, and hopefully this will be very intuitive. Hopefully everything I'm telling you is like, oh yeah, I guess it'll kind of have to happen that way. So anyway, here we go. Step 1, codon recognition. Obviously that would have to be the first step, right? That tRNA with its very specific anticodon and its very specific amino acid, it recognizes the codon and says, oh, yep, that's me. Right? So it comes in bringing in its amino acid, right? That's step 1, is codon recognition. And again, you can see here I've written that this is happening in the A site, but I'm not going to ask you about that. It's there. I want you to understand it, but I'm not going to ask you that on the exam. You don't need to know that it's the A site where the things are coming in and the P site where the other stuff is happening that I haven't told you about yet. But yeah, that's step 1, codon recognition. So I will see. I know it's Monday. Everybody seems a little bit tired, and this is a little bit complicated. I want to see if anybody can guess off the top of their head. Once this thing, picture it, once this thing gets here, what do you think might happen next? Think about the end goal here. Any guesses what might happen? If you get it wrong, I won't blame you because it's kind of vague. We'll see if anybody can guess what might happen next. Not the name of it, but like physically. So can you figure out what might happen? Nope. All right, sorry. Next is peptide bond formation. So now that you have these things right next to each other, and remember the end goal is to add an amino acid to this growing strand of amino acids. Obviously, the next step, once they're together, is to make those peptide bonds, right? Those covalent bonds between this new amino acid and that amino acid. And again, there's names. I've mentioned this. The P site is where the growing strand of amino acids is. The A site is where the new amino acid is coming in. I'm not going to ask you about that on the exam. Also notice too that what's happening here is that this growing strand on the P site is being covalently bonded to this new one on the A site, not the other way around. That'll be slightly important on the next step. So speaking of which, visualize this if you will. What's happened now, try to visualize that this is now attached over here, right? So now you have on the A site, you've got this strand of amino acids. On the P site, you have this tRNA that is now empty because it gave up its amino acid. So any guesses what the next step might be? Not the name of it, but what actually happens. Think about this too. We need to keep moving, right? We're not done yet. We only have 1, 2, 3, 4. We only have 5 amino acids. Let's assume we need 100 in total. So what do you think must have to happen next for this process to keep moving? Again, if you can visualize it, this thing is now without an amino acid. You just have a tRNA, empty tRNA sitting there. And now you have, on the A site, you have a tRNA with a long strand of amino acids. So any guesses? Nope, that's okay. Translocation is next. Obviously, if this thing has to keep going, then, well, we got to keep things moving, right? So that tRNA that no longer has an amino acid, it leaves, right? Don't need you anymore. Go away. Likewise, that tRNA that's sitting there in the A site, I'm going to ask you about that on the exam, it's in the wrong place. it's blocking any new amino acids from coming in because that's the A site is where they come in. So its job now for translocation is to get out of that spot and go into the newly vacant spot where the other tRNA just left, the empty tRNA. So again, I hope this is very intuitive for you. It might be a little bit hard to memorize, but I'm hoping that's very intuitive. All of that, it just has to work that way, right? It just makes sense. So again, that thing leaves, this thing moves over. Now, and even though you can't see it, now the A site is empty again, and now we can start the whole process, right? Now that this thing is over here with this in the P site with this long strand of amino acids, and the A site is now open. Now, well, then we can just go back to the first step and bring in a new tRNA with a new amino acid, and the whole thing starts again. Pretty simple way we're describing it, right? And again, I'm hoping that's very intuitive. Obviously, you're going to have an open site for a new one to come in. Obviously, you're going to have to covalently bond, right? You need to make those peptide bonds. Obviously, once that happens, you need to get rid of the old tRNA because it's useless now. You need to move this so that the opening site is open again, right? So bring in the new tRNA. I don't think this picture is from your textbook. I don't remember what the picture in your textbook looks like, but I do remember the first time I saw your textbook thinking that the old textbook had a better version, so there it is. Any questions? That's it for elongation. Now let's talk about the last one. This is nice and easy. Termination, just like transcription ends with termination, same here. The only thing different that might be a little bit hard to differentiate on the exam to memorize this, not that. So remember with transcription, it all ends at the terminator, which is nice and easy, right? Termination happens at the terminator. Here, it's a little bit more complicated, not even that complicated, but so it stops at the stop codon. So the stop codon is the terminator of translation, so to speak, right? So the terminator in transcription is where transcription stops. The stop codon in translation is where translation stops. And that for me was hard to memorize as a freshman. Because really, even transcription, when you talk about transcription, the terminator is it's a set of codes. So it was hard for me to remember that because technically you're still dealing with codons when you're dealing with transcription, but they named it the terminator, not the stop codon. Now, there's 3 different stop codons, and you don't need to know either one of them. But there you go, just in case. UAA, UAG, and UGA. That's not the University of Georgia. But yeah, those are the 3 stop codons. And you already learned about that, actually. When we talked about the 20 different amino acids and all the 64, when we're talking about the language, right, the amino acid language, the nucleic acid language, right? In that language, every word has 3 letters in it. There's only 4 different letters to choose from, A, T, C, or G. Or for the RNA, A, U, C, and G, right? And then we said, so that means there's 64 words in that language. 61 of them say, bring me a specific amino acid. And 3 of them say, stop. So there you go. We'll point this out too. So I've told you this before. There's going to be a question where I give you a DNA code, and I'll say transcribe it, right? So then you make an RNA code out of it. Or I might say translate it, so then you have to transcribe and then translate. Anyway, my point here is when I do that, I just do random letters. So if I accidentally make a stop codon in the middle of a strand of RNA, you can ignore it. So don't think, is this a trick question? Because he's got UGA right here in the middle. That should be the end of it. So ignore that if I accidentally make that mistake. I'll try not to. Anyway, any questions about this? Well, no, one more thing, sorry. Stop codon is the thing that says this is it, we're done with it. But then what happens? This is the easy part. Everything just goes its own way, right? The tRNA goes away. The amino acid goes away. The small subunit goes away from the large subunit. The mRNA goes away. You could ask this, though. Again, this picture's not from your textbook. For extra credit, you could look into what happens. I mean, I'm going to tell you what happens to the polypeptide, but what happens then to the tRNA? What's going on there? It's like it's done now. Does it just get recycled or is it digested? What happens to all that stuff? Anyway, or the mRNA for that matter, right? We're done reading the mRNA. So what happens? Is it just sit there floating around in the cytoplasm so it can be transcribed again? Or does something happen to it? All these things you could recover for extra credit. Any questions about that? Next word for attendance is Navy. For some reason, today is National Navy Day. I don't understand it because their birthday was earlier this month. So it makes sense to me to have Navy birthday on the Navy Day, but I don't make the rules. So real quick review. We got less than a minute, so I won't do that. We'll review when we get back. That'll be a good time to do it to refresh your memory on Wednesday. So that's it. Don't forget to send the words. Don't forget to include one question about the material presented in this lecture. Yeah, that's it. I'll be in my office and online for office hours. Have a good week.